Okay, welcome everybody. For this is the inaugural Shu and Casey Chen lecture in biomedical engineering. So first, I just want to say a little bit about Shu and Casey. In your book, there's a lot more information. They actually met at National Taiwan University, where they both received medical degrees, been married for, I don't know how many years, 60, 70 years. Um, and of course, Shu is a Columbia person, right? He actually got a first assistant professor's job here in physiology way before there was biomedical engineering department. But he became really a pioneer and one of the founding fathers of biomedical engineering in the country. He went together with Richard Scalak. They left Columbia, started the program at UCSD, which of course has been world class. And if you look at the awards that Shu Chen has received, they're amazing, right? He's a member of all the national academies. He's won prizes that span across the entire world. He's won the National Medal of Science, highest award in the United States for a scientist. And but what's really impressive about Shu Chen is what a modest, nice guy he is, right? So all of his success, everything he's done for the community, he's still, a humble person. And he's still working today. He's 93, 93? And apparently he's just gotten the record for not just the longest continuous RO1, he just got it, but the oldest person ever to have an RO1. So this is somebody who's committed to science. He's committed to biomedical engineering, and it's really our honor as a department that he wanted to endow this professorship. This lectureship. So with that, the inaugural Shu Chang Distinguished Lecturer is Hanju Zhou. And in fact, when we asked Shu Chen, did he have any recommendations about who should be the inaugural lecturer? He thought about, no, you guys do it, whatever you want. And then he called us back saying, no, I want actually Hanjoong to be the inaugural lecturer. And Dr. Zhou is also an amazing individual. He's actually the distinguished chair professor, Coulter distinguished chair professor and associate chair of biomedical engineering at Emory and Georgia Tech. He is a professor of medicine at Emory and director of their cardiovascular biomechanics T32. So once again, not just research, but very much Dr. Zhou's engaged in training. He also has a very impressive set of honors and awards, fellow of AAAS, BME Society, AHA, American Physiology Society, and is also a very humble individual. Had dinner with him last night. It was really fantastic. Is doing some great work in looking at studying how blood flow regulates vascular endothelial cell function. And he also is very engaged in different societies, including have been former vice president of the Korean American Scientists and Engineers Association. So let's welcome Professor Zhou.
Or thank you so much for the really kind invitation. And this is especially honorable to be invited by you with my role model throughout my entire research career since I was a grad student. And I always wanted to be like you. So I just found out that Shu really recommended me for this lectureship. So thank you, Shu and Casey, for the invitation, and the faculty and staff, students. It's a great honor to be here to introduce the work that I've been working. So what I'm going to do today is actually the work that Shu and I have been in the same field. I think probably Shu recommended me because I was trying to be like him, and sometimes we would publish a paper just ahead of him. So maybe. So what I'm going to talk about today is how flow regulates vascular endothelial cells, how flow reprogram endothelial cells, and how this plays a role in the development of atherosclerosis. So what I try to do is that I will introduce the concept of mechanobiology and how we use this mechanobiology to treat diseases as a mechanomedicine. And I will use one of the examples of Hanjoong in the second half of the story to talk about how we use this mechanobiology knowledge towards mechanomedicine to treat atherosclerosis. So atherosclerotic disease is the #1 killer in the world. Atherosclerotic disease include heart attack, ischemic stroke, and peripheral artery disease. And there are a lot of therapeutics available, such as lowering cholesterol or hypertensive drugs that really help reduce cardiovascular disease in general. Yet, this continues to be the #1 killer. What that tells us is that currently available drugs and therapeutics are not enough to prevent these cardiovascular disease. So what is left? So this is something that we call a residual risk factor, and we are suggesting that flow is one of the critical residual factors that we could go after. And I will tell you why we think this flow-dependent gene is important, because as you will see, that atherosclerosis preferentially occurs in areas, arterial regions where flow is disturbed. And so that's why we are going after this. So briefly, for introducing atherosclerosis, atherosclerosis is a arterial thickening caused by a accumulation of lipids, fat, and blood cells, especially immune cells, muscle cells, that occur in people over decades. And if this goes on and causes a real problem, sometimes these plaques rupture, and that causes heart attack if it occurs in coronary arteries, or ischemic stroke if it occurs in the carotid arteries or in the brain arteries, or peripheral artery disease if it occurs in the peripheral arteries. Overall, this is just a brief overview of how atherogenesis occurs. Again, this is a arterial wall model. Endothelial cells are in the middle, and this is where blood is. And what happens is, when you have a risk factors, such as high cholesterol or flow disturbance, endothelial layer becomes inflamed, and this causes the permeability of endothelial cells to LDL, carry cholesterol, increases, they come in, they get oxidized, and additional signal is generated to inflame endothelial cells, to bring in immune cells, such as monocytes, to the arterial wall, to take care of the problems. But then what happens is, on the chronic inflammatory conditions, these monocytes become macrophage, they take up oxidized LDL, they become foam cells, macrophage-derived foam cells. And these foam cells are really what's causing the plaque. Now, as you will see from this lecture, macrophage used to be the main cell type that would lead to plaque, but now more recent data says that some muscle cells also cause plaque development, and as you will see today, endothelial cells also become a plaque and part of the plaque developing cells. So why distal flow? If you look at where atherosclerosis occurs, it always occurs in branching points or curved arteries, such as coronary arteries or carotid branches. So for example, if you look at carotid branch here, this is where plaque is, and this is a common carotid artery that is in our neck. But this straight portion of the heart is exposed to stable flow, and they are not developing atherosclerosis. But in this area, where you will see flow is circulating, along this area is where flow is really disturbed. It's characterized by low and oscillating shear stress. And this is the area where we begin to see atherosclerosis. But the field did not know whether flow disturbance actually is causing atherosclerosis or not. And that's what my lab's major contribution is. So before I get into more details, just as an introduction, if you look at the artery or blood vessels, there are two major biomechanical forces. One is a shear stress that is caused by a dragging force of a flowing blood. That's a tangential dragging force that is on the endothelial layer, and that is one of the key biomechanical factors that we will discuss today. But there are blood pressure caused by a volume of the blood, and this also causes a circumferential or hoop stresses. And when there is this mechanical force, endothelial cells are really designed to detect these mechanical forces. And there are dozens of mechanosensors that are on top of endothelial cells. Some are in between cells, and these include adherence junction proteins such as beta-catenins, B-caterin, PCAM1 complex C in the cell-cell junction, but also at the cell surface there is a G-protein coupled receptors, piezo-1, glycocalyx, primary cilia, NOTCH1, plexin B1, and caveolin. These are all considered to be a mechanosensor. And some of them are at the bottom of the cells, such as integrins, and also cytoskeleton is also part of the mechanosensing mechanotransduction complexes. Once there is a shear stress, depending upon whether the shear stress is uniform, unidirectional, or multidirectional, it will cause a different type of signaling that will eventually lead to changes in endothelial cell phenotypes, such as endothelial cells become mesenchymal cell types, called endothelial cell to mesenchymal transition, or cells inflamed, angiogenesis, permeability increases, cell alignment changes, and various changes occur within endothelial cells that are believed to eventually lead to atherogenic development. So when we were actually trying to understand the effect of flow in atherosclerosis, actually there was no specific animal model to test this hypothesis, whether distal flow can actually induce the disease while stable flow can prevent it. So we developed this model known as a partial carotid ligation model of atherosclerosis. This is a mouse model. We are using two carotid arteries in the neck. As I told you in previous slides, in the common carotid artery area, they are exposed to a stable flow. If we don't do anything, they are always exposed to stable, unidirectional, pulsatile flow that goes toward the brain. And that area is protected from atherosclerosis. So we changed this flow pattern from stable flow to low and oscillating flow by just ligating 3 out of 4 downstream branches that goes into the face and the brain. And we left one branch open, and that caused a low and oscillating flow in the left common carotid artery. So with this model, then we try to develop atherosclerosis. If we don't give them hypocholesterolemia conditions, they don't develop atherosclerosis. You need a second hit. So in this condition, what we did was we used a ApoE knockout mice model, or we can actually cause a condition to remove LDL receptor knockout, then plus hyperdiet feeding. This will lead to hypocholesterolemia in whole mice, but only in the left side. That's where we did a carotid ligation surgery, where flow is disturbed. Within 2 weeks, we can develop atherosclerosis in the left side, but not in the right side. Even though they are in the same animal, one side is under stable flow, this side is a distal flow plus hypercholesterolemia. And if you cut in the middle to look at the inside, you can see in a time-dependent manner, atherosclerosis develops. By 2 weeks, it begins. By 4 weeks, it develops advanced features of atherosclerosis. At the same time, if you look at the right side, exposed to stable flow, even though cholesterol is super high, they don't develop atherosclerosis. So this really provided the proof that stable flow protects, distal flow induces atherosclerosis in the face of conditional hit, such as hypoglycemic conditions. So based on these, and this model has become now a standard model in our field, and a lot of labs around the world use this model to understand how distal flow cause these atherosclerotic phenotypes. So distal flow is sensed by endothelial cells, then it causes a endothelial cell nitric oxide synthase uncoupling and reactive oxygen species activation that causes endothelial dysfunction. And during this time, the immune cells then begin to infiltrate to the arterial wall to cause this atheroma formation. And by 4 weeks, you can see advanced lesion forms. So really, this is really so remarkable, very fast change by just changing flow conditions. And this is mediated by changes in genome, epigenome, and metabolomic-wide changes in the arterial wall by distal flow, especially in endothelial cells in vivo. So to understand this mechanism, we and others have done a lot of omics studies using our model. So, for example, using mouse partial cartilagation model, we have carried out single-cell RNA sequencing, or bulk RNA sequencing, or messenger RNA arrays, microRNA arrays, and DNA methylome studies and metabolomic studies. Throughout these, using these omic studies, we have understood a lot of the mechanisms. We identified a lot of the targets that change under flow distal conditions. And then we used a lot of a big data analysis method, single cell analysis method, now machine learning, and we are now moving toward a AI-based method to predict which gene changes or protein changes are responsible for atherosclerosis development. And we spent a lot of time to go after individual genes and understand the mechanisms, how flow-sensitive genes can then change endothelial function and eventually atherosclerosis development. And from using these type of cellular as well as in vivo studies, we have identified several genes that play critical roles as a flow-sensitive gene that regulate endothelial cell biology as well as atherosclerosis. And I will talk about HEB1 as one example in the second half. Now, also, after we develop these therapeutic gene targets, then what we do is the delivery. So we want to do a targeted delivery using various methods, using lipid nanoparticles or ultrasound-based methods. So this is what we call a going from mechanobiology to a mechanomedicine. So to understand this gene changes a little bit better, I'll give you some examples of what we have done. So we have carried out single-cell RNA sequencing and single-cell ATAC sequencing to figure out how transcriptome or epigenomes change in response to flow conditions. So using our partial cartilagation model, after the partial cartilagation, 2 days or 2 weeks later, we collect RCA exposed to stable flow or LCA exposed to distal flow. And then we did a single-cell RNA sequencing or ATAC sequencing. And we then analyzed the data. This is a UMAP analysis showing how individual endothelial cell populations change along the stable flow conditions at 2 days versus 2 weeks, or in response to distal flow conditions at 2 days and 2 weeks. What you can see is that on the stable flow conditions, we have 4 different types of endothelial cells. They are healthy endothelial cells. But as they are exposed to distal flow, they begin to change cell transcriptomic profiles. And if you actually look at after 2 weeks, we can see that some of the cells are really new type of cells are emerging. So for example, E2 is a prototypical healthy type, E8 is a prototypical pro-atherogenic types. And if we do a lot of analysis, for example, if we do a RNA data trajectory analysis, we can see healthy endothelial cells change in one way to mesenchymal type of cells. So this is a endothelial cells in response to distal flow transitioning to either smooth muscle-like or fibroblast-like cells. That's a endo-MT, endothelial cell to endo-MT. And endo-MT is well known to be important in atherogenesis. On the other hand, we found something new. That is, endothelial cells, healthy endothelial cells begin to change toward immune cells. This was just a distal flow alone. Distal flow alone was stimulating endothelial cells to begin to switch toward endothelial cells, but it didn't go all the way. So we called it a partial endo-IT, endothelial cell to immune cell transition. And so that was a in vivo data. So we wanted to see a in vitro conditions to see whether if you have a pure endothelial cells and culture it in under a stable flow conditions using unidirectional laminar shear or oscillatory shear conditions to simulate distal flow conditions. After 1 week, you can see the endothelial cells align to the direction of the flow by the stable flow, but oscillatory shear lost that alignment. And if you look at the gene markers, you can see the markers of immune cells go up under distal flow conditions, or endo-MT markers also go up in response to distal flow conditions, demonstrating that what we saw from in vivo conditions actually can occur with just a pure endothelial alone in vitro, demonstrating that endothelial cells have the capability to switch to different cell types under flow conditions. So based on this, we initially determined that distal flow alone can change, induce changes in epigenomic pathways and transcriptomic pathways that lead to increased pro-atherogenic gene expression types that lead to increased endothelial inflammation, endo-MT, and partial endo-IT. And we call this as a collectively FIRE, flow-induced reprogramming of endothelial cells. So this is a partial FIRE caused by distal flow. So after doing this experiment, then we next asked the question, can we actually induce a full FIRE by combining distal flow plus hypocholesterolemia conditions? And so we did a similar experiment. Again, we are using a black sex mice. And to cause hypocholesterolemia conditions, we can just inject with this PCSK9, AAV-derived PCSK9. This will go in and delete LDL receptors in the liver. And if we give them Western diet, then it causes hypocholesterolemia conditions. Using this hypocholesterolemia conditions, then we did a partial corrected ligation surgery to cause a distal flow only in the left side. So we have a control group, distal flow alone group, high cholesterol alone group, or distal flow plus hypocholesterolemia groups after 2 or 4 weeks, only in conditions where you have distal flow plus hypercholesterolemia causes plaque development. So now we took these arteries and did a single cell RNA sequencing. So these were done over about 100 mice, and at the end we have about 100,000 single cells analyzed, and we projected it together in this UMAP analysis. And we can show that there are about 5 different types of endothelial cells, 3 different types of muscle cells, 3 different types of fibroblasts, and also 5 types of macrophages, and dendritic cells, T cells, and NK cells, and B cells, etc. So all the cells that you would expect to see in atherosclerotic plaques are represented in this single cell RNA sequencing data. And the reason that we know what they are is that based on canonical markers of individual cell types. So based on that, we can be sure that we are calling the right cell types. So to understand how endothelial cells change under different conditions, we then just looked at endothelial cells alone. So these endothelial cells, these are whole endothelial cells in the entire groups, but these are then divided into individual groups. Control conditions at 2 days, 2 weeks, 4 weeks, or hypocholesterolemia conditions at 2 weeks and 4 weeks. Distal flow alone for 2 days, 2 weeks, and 4 weeks, and distal flow plus hypercholesterolemia at 2 weeks and 4 weeks. If the cells, individual cells look about the same, they should have the same color. So under control conditions or high cholesterol conditions, endothelial cell shape is about the same, except for a transient endothelial cell to mesenchymal transition at 2-week time point, but at 4 weeks we lost that. In contrast, flow disturbance actually changes a color. That means endothelial cells are transitioning to a different type of cells. So as you can see, there are red showing up. These mean that cells that are pro-inflamed or undergoing endo-MT are showing up by just distal flow alone. If you combine distal flow plus hypercholesterolemia, you can see there is a dramatic increase in red type of cells. These are cells that show immune cell as well as foam cell features. The foam cells that produces plaque. And these are all the markers that we use to assign individual cell type transitions. And I'm going to skip smooth muscle cells and macrophage data to save time and just focus on endothelial cells. But if you are interested, I'll come back and answer any questions. So again, this is the total number of cells. And again, I divided it into 10 different conditions that we have done. Control, high cholesterol alone, distal flow alone, and distal flow with hypercholesterolemia. And you can see that there is a dramatic change in cell distribution. So you can see in cholesterol conditions, in hypercholesterolemia conditions, actually, there's not much difference between cholesterol and high cholesterol conditions, which was a surprise to us a little bit. But distal flow, you lose this ostrich-like shaped cell distribution. They lose the hip, and the cells just distribute all over the place. That means cells are really changing their phenotypes. But if you look at this distal flow with hypercholesterolemia, you can see the ostrich shape became a horse-like shape. So it just completely transitioned from ostrich distribution to a horse type of distribution, meaning flow plus hypercholesterolemia dramatically changed how the cells are, the individual cell phenotypes. So that was all single cell RNA sequencing data. It's great. The single cell RNA sequencing data is a wonderful starting place to form the hypothesis. But you need additional validation. So to get additional validation, we focused on endothelial cell transitioning to other different cell types, reprogramming of endothelial cells. To do that, we used these confetti mice. These confetti signals such as GFP, YFP, RFP, CFP can be expressed in this model only in endothelial cells in response to tamoxifen induction. So if they express these confetti markers, they are endothelial cells, generically encoded endothelial cells. Then we used these mice. We fed them with the hypercholesterolemic conditions and did a distal flow induction. And after 4 weeks, you can see that in these mice, they all develop plaque development. And then we looked at whether these plaques show the evidence of endothelial cell reprogramming by looking at the markers, the canonical markers of inflammation, endo-MT, endothelial cell to immune cells, or endothelial cell to foam cell markers. So what we did was we did a longitudinal sectioning to look at the entire length of the carotid artery. So this is again our partial carotid ligation model after 4 weeks. The right side is exposed to stable flow, whereas left side is exposed to distal flow, and they develop atherosclerosis. And we can cut it entire lengthwise, and you can see lumen is here, and this is a 2 cuts of the left common carotid arteries, and you can see the plaques in here. And this is a close-up view. So to look at where endothelial cell is showing inflammation markers, we use VCAM1 or ICAM1 as a marker. This is the lumen. Here is endothelial cells marked with a red or green. These are the confetti markers. So they are endothelial cells. Not all endothelial cells are marked in this manner. Up to about 40% of the endothelial cells are marked with confetti markers. Anyways, these confetti positive endothelial cells express VCAM1 or ICAM1. So endothelial cell inflammation occurs. That was not a surprise. We knew this was going to happen. The next was endothelial cell to mesenchymal change, endo-MT. Again, this is a low magnification view, and this is a high magnification view. APTA2, SNE1, and CNN1 are canonical endo-MT markers. You can see these confetti positive endothelial cells express markers of APTA2, SNE1, and CNN1 in endothelial cells. So this proves that endo-MT occurs in response to distal flow plus hypercholesterolemia. This is a endothelial cell to immune cell markers. So again, CD68, C1qa, C1qb, lysine, these are all immune cell markers that is expressed in endothelial cells, genetically marked endothelial cells, demonstrating that endo-IT occurs. The next one was endo-FT. So this is, again, looking at markers of SDP1, LDL-S3, TREM2, and ODP, lipid staining in endothelial cells. So again, using these various markers of endo-FT, endothelial cell to foam cell, we can demonstrate that endothelial cells indeed become a foam cells. So based on this, what we have concluded was that there is a two-hit hypothesis. As I told you, atherosclerosis requires distal flow plus high cholesterol. So what distal flow does is that it causes the initial inflammation of endothelial cells, and it also causes a LDL permeability to go up and increases these initial foam cell development. But it's really at the minimum level. It doesn't really generate a lot of plaques. But if you then add cholesterol to this condition, then endothelial cells become fully transitioned toward immune cells, and they become foam cells. And some of them actually seem to migrate in. So we think that distal flow is the initial trigger. It's a pilot light. But the pilot light alone does not cause a whole forest fire. You need cholesterol, high cholesterol. High cholesterol is the fuel that really starts the whole forest fire. So in combination of the distal flow plus hypercholesterolemia, then we can actually develop atherosclerosis. And the changes that occur by distal flow at the level of endothelial cell genes are the gene targets that we are actually going after. So one of the genes that we identified from our single cell RNA sequencing data was HEB1. And again, we have published many other genes, but I'm just going to talk to you about this one gene that we recently published. So this gene is reduced by distal flow. Stable flow increases, distal flow decreases. So we thought that this must be a protective gene. So this HEB1 gene, it's known as a heart of glass. Initially, this gene was identified because if you knock out this gene in zebrafish, heart becomes big and they break. The heart becomes like a glass. So the initial investigators named it as a heart of glass. So we knew this gene would be important in cardiovascular disease, but nothing was known about its function in endothelial cells. So this HEB1 gene is a one single transmembrane gene. It is highly glycosylated. So think of it as a huge tree sitting on top of a hill. When the wind blows, this big tree will just sway by the wind. That's what we thought, and maybe that's how it works as well. Anyways, this one has a transmembrane domain, and in the C-terminal, it's inside, it binds to some of the proteins known as a CCM complex and RACID1. and they play an important role in transmitting the signal. So then first we just validated whether a single cell RNA sequencing is correct by looking at our partial character ligation model. You can see the green signal in endothelial layer is high in RCA, meaning stable flow conditions have expressed high level of HEC1. But in response to disturbed flow, the green signal is lost. That means HEC1 is lost. And we can do that by various ways, by both this RNA scoping as well as qPCR to further validate the data. In an in vitro study using human aeric endothelial cells, we studied what would happen if we expose them to either stable flow or disturbed flow conditions using our cone and plate model. So what we found was that within 24 hours, HEC1 protein increases in the cytosol. Another surprise was that HEC1 is not only expressed in endothelial cells, actually it gets secreted into the media. And it actually circulates in the blood, and actually we can measure it. And although I'm not going to show you the data, in people with hypertension and cardiovascular disease, this level is reduced. Suggesting that this is playing some kind of a protective role. So we did some additional detailed studies to look at how protein gets expressed in a time-dependent manner and how it gets really released into the conditioned media in here. So again, within 24-hour period, HEC1 expression goes up by stable flow conditions, and you can see that it's secreted into the conditioned media in a time-dependent manner. And we then looked at immunocytochemistry to see whether HEC1 protein actually changes its location by flow. And as you can see, after 24 hours, the red is HEC1 protein staining in human aeric endothelial cells. You can see in steady conditions, it's diffusely located. But after shear stress for 1 day, you can see they move toward cell-cell junction areas. But in response to disturbed flow conditions, HEC1 protein expression is reduced. So we looked at its movement in a more acute manner, and you can see that compared to control, within as early as 2 minutes, we can actually see that it's moving toward the membrane junctions and actually accumulates in junction area. So it moves downstream and it accumulates in the cell-cell junction. So this is initially what we found. Under stable flow, under disturbed flow conditions or steady conditions, HEC1 is diffused and expression is reduced. But in response to disturbed flow conditions, HEC1 is produced and it comes out and it moves downstream towards cell-cell junction. Some of the HEC1 is also released into the media. That means some of the HEC1 produced from endothelial cells circulate in the blood. Next, we tested its function. So we, using human aeric endothelial cells, we first measure the endothelial cell inflammation by measuring how much monocytes bind to endothelial surface after they are exposed to either steady conditions or stable flow conditions. As you can see, the amount of monocyte binding to endothelial surface is reduced. That's a known effect. A stable flow reduces inflammation by preventing monocyte adhesion. But this reduction is reversed if we knock out HEC1 gene, meaning HEC1 is providing this protective anti-inflammatory function. Similarly, permeability is also reduced by stable flow, and that is also lost if we remove HEC1. Tube formation, angiogenesis response is also reduced by stable flow conditions, but this also goes back if we knock out HEC1. And migration is similar. So what this shows is that the function of stable flow, the anti-atherogenic function of stable flow, is mediated by HEC1. If we remove HEC1, they lose that protective function. And we then did a lot of cell signaling studies that I will skip the details of the cell signaling, but basically we used a lot of single siRNA techniques or inhibitors to establish this following pathway. So if you expose endothelial cells to stable flow, and if you knock out siRNA, the subsequent pathway, the activation of NEKK3, NEK5, ORG5, which then goes into the nucleus to activate NAF2 transcription factor and activation of KLF2 pathway. This is a pathway that is regulated by this sequence. Again, due to time, I'm not going to get into all the glory details of the experimental methods. So then what we did was we wanted to see its in vivo function. So we knocked out HEB1 in endothelial cell specific manner by using CRISPR-based methods. So once we knock out HEB1 in endothelial cells, first thing that we measured was permeability to LDL cholesterol. If we inject LDL cholesterol, that usually goes a little bit to the LCA area, the flow disturbed area. This is significantly exaggerated if we remove endothelial cell specific HEB1, meaning that HEB1 loss is really causing permeability barrier loss. We then tested its effect on atherosclerosis. This is a wild type condition, and this is a HEB1 endothelial cell specific knockout. You can see more atherosclerosis occurs if you lose HEB1. If you look at the details in comparison here by looking at the longitudinal sections here, you can see even though actually the size of the overall plaque may not be so high, you can see the quality of plaque is dramatically different. So compared to wild type, where it's just the thickening of the plaque, you can see a lot of macrophage inflammation and a lot of empty spaces. The necrosis actually occurs within this conditions. So this is really showing different pentachrome staining to demonstrate this further. So again, this is a plaque developing in the wild type conditions, but this is exaggerated in the condition where HEB1 is knocked out. So the really important feature is that this causes what we think of as a vulnerable plaque feature, those plaques that could rupture in patients. So that is characterized as a thin cat fibroatheroma or TIPFA. So we can see compared to this thick fibrous cat here, cat is very thin and fibroatheroma is really big, and necrosis occurs, and hemorrhage occurs, intraplaque hemorrhage occurs, and this is really representing an advanced vulnerable type of plaque occurring when you remove HEB1. So then we did an additional study just without partial creatinine ligation models, just giving them high-fat diet for 2 months. And as you can see, compared to wild type, where it shows red staining here, that's atherosclerosis, but there's a lot more atherosclerosis occurring within 2 months. If we do this at 4 months, you can actually see much more clear difference. This is a liromate control. This is a HEB1 endothelial cell knockout. Again, just a high-fat diet alone for 4 months. It really demonstrates a huge effect, exaggerating effect of HEB1 knockout on atherosclerosis development. This effect does not change cholesterol levels or overall plasma lipid levels in all males and females. So this HEB1 dependent changes is independent of any lipid changes. Another interesting thing that we found was that, as I told you, HEB1 is made in endothelial cells and it goes into the circulation. So we thought that it may have some additional circulatory effect. So we measured blood pressure. And we can actually see, just as soon as we knock out endothelial cell HEB1, we can see the blood pressure rise. But now if we then add them additional condition of a hypocholesterolemia conditions, we can actually see blood pressure even further rises. And actually, the people who are studying hypertension, it's really rare to knock out individual gene and see these natural hypertension conditions. So some of our colleagues are very excited about our finding. So we are now trying to figure out how hypertension occurs. as well. We think that the circulating HEG1 may have something to do with it. We also looked at human patient samples in the coronary arteries who have developed a heart attack. So these are human coronary arteries that have either a little bit of plaque to a really advanced level of plaques. And if you stain for HEG1 levels shown in red in endothelial cells, you can see in less diseased conditions versus highly diseased conditions, HEG1 protein level is reduced.
I didn't understand that.
Okay, sorry. Sorry, never listened. So, in summary, what we found from HEG1 study is that on the distal flow condition, HEG1 level is reduced. And when HEG1 is reduced, this leads to a inhibition of this important KLF2-4 pathway in response to the stable flow conditions. This somehow moves HEG1 away from diffuse conditions downstream towards cell-cell junction, and this causes this pathway, this MAC pathway, ORF5 pathway, to be activated to induce KLF2 and 4 induction. KLF2 and 4 actually is a master transcription factor that plays a critical role in protective function by flow conditions. So we think this is actually the first demonstration of one of the critical receptors at the top can regulate KLF2-4 induction by shear stress. And some HEG1 gets released, and this we think has some circulatory effect, some systemic effect. So under these conditions, especially on the distal flow conditions, with hypercholesterolemia, this causes endothelial inflammation, permeability change, tube formation and migration, and hypertension. And this is caused by a endothelial cell 5 flow-induced reprogramming. And the genes that are involved in this pathway is going to be really critical to develop additional anti-atherogenic therapeutics. So in summary, my lab has been really using mechanobiology approaches to understand how flow regulates endothelial cell biology using various omics approaches and doing a detailed mechanobiological studies. And also based on this, we are developing new therapeutics that target flow-sensitive genes as a residual risk factor targeting therapeutics. And also we are working on delivery mechanisms. So again, this work is done by a talented group of students, including one of a Columbia BME student. Christian Park received a bachelor's degree from Columbia, so he has done all the single-cell RNA sequencing studies. And in Camargo and Kangbaek, Yerin Kim, Dongwon Kang, Michael Clark, and Jay are the key members who have carried out the studies that I presented here. And these are my colleagues at Emory and my funding sources, and these are my team members, where when we are not in the lab, we go with the flow by going downstream. So thank you so much for your attention.
Thank you, Professor Joe, for such a wonderful talk. Questions? I'll start with one. Is there any frequency dependence of that oscillation and then the buildup of the plaque, or?
Yeah, there may be a frequency dependency. We haven't really characterized it. But one of the major mechanical force change is the directional change. Whether you have a unidirection or multi-direction, that is one of the most important determining factor. The second thing is the magnitude. So low magnitude and oscillating patterns. This is the key point.
Don't be shy. Come on.
Man.
Thank you. I really enjoyed the talk. I thought the endothelial to immune transition was really interesting, and I was wondering if you could maybe theorize about why the body does this, and whether there's other contexts, maybe physiologic contexts even, where this type of transition occurs.
Yeah, so again, this was a surprise to us that endothelial cells become immune cells. But there was some literature for many years previously that endothelial cells perform immune cell-like functions. So endothelial cells undergo inflammation, and but it was unknown whether endothelial cells actually transition towards immune cell-like cell types. We think that this is actually one of the most crucial novel area of study, and I think these chronic inflammatory phenotype under distal flow conditions, exacerbated by hypercholesterolemia conditions, is one of the most crucial areas that we need to look at. And if we can bring back or reverse this endo-IT phenotype, we think that that would be an important therapeutic area.
Yeah, it's really fascinating. So are there other parts, I mean, the flow is sort of a very particular thing that's happening, and so are there other endothelial areas that are also having this, transparent heart protein that is playing a role, or is it somehow there particularly because of a problem with the flow that would cause the endothelial cells to act strangely because it's sort of a very particular state that they're in there in the blood vessels?
Can you clarify your question a bit more?
Sorry, no, I'm just wondering if these processes that you're finding are very unique to blood flow and flow conditions, or if they show up in other conditions as well.
So actually, for example, HEG1 expression can be affected by not only by flow conditions, but a lot of other systemic proatherogenic conditions. So we tested the conditions, high cholesterol conditions, or cytokines, TNF-alpha, interleukins, or angiotensin 2, LDL cholesterol, and diabetic conditions. They all actually reduce the level of HEG1. So we think that HEG1, although we focused on just studying flow, but there are additional factors that could regulate these genes.
Present